How many lectures do we have? 6?
6.
1, 2, 3, 4, 5, 6. This was introductory material. What is the nervous system? What is the difference between the PNS and the CNS? What's the difference between motor and sensory? And then we reviewed the action potential stuff that we did in the previous year. This is action potentials. And then we ended with a little preview with the beginning of the CNS. Mostly just action potentials. CNS, PNS, eye, ear. We also threw in general. And then today we're going to do ear. And then in addition to ear, we'll do taste, but that's a very small point. And then olfaction, we'll just throw in at the end, but we've already done that as well. So really, ear, eye. So that's how many questions you're looking at. Right? It's going to be statistically pound for pound. 1/6 of it is going to be brain. A little more than 1/6 of it is going to be, or about 1/6 of it is going to be action potentials. Okay. This is really mostly cranial nerves for the most part. Basic structure of a nerve, 12 cranial nerves. 1 or 2 questions on sympathetic versus parasympathetic, but it's not a ton. And then a whole bunch on eye, a whole bunch on ear. So, does that answer your question?
Yes.
Okay.
But that doesn't help me answer yours.
No, not necessarily. It shows you where you needed to tune this stuff. This is, eye and ear are really just largely anatomy. We're not doing a ton of physiology, but there'll be 1 or 2 things, right? For example, we said you got to trace the path of the eye, or the path of light through the eye, rather. We'll do something very similar with here. Right? This is mostly going to be cranial nerves because that's what we focused on that lecture.
What was that second part? I have a hard time.
Here?
Yes.
Action potentials, and then our very beginnings of the CNS. What we did really on this day with the CNS was the ventricles of the brain, the production of cerebrospinal fluid and its circulation.
Can you go over the plexus?
Which one? There are a bunch of them.
Yeah.
Are you talking about the choroid plexus of the ventricles, or are you talking about the nerve plexus?
I think it's just looking at it, I just like.
Okay. So when you're talking about the ventricular system, right, you've got a lateral ventricle that's kind of shaped like this horn, right? And then you've got the third ventricle, and then you've got the fourth ventricle, and then you've got down into the spinal cord, right, the central canal. That's a horrible picture, but that's the best I can do on the fly. So there are little beds of capillaries. You can go up and look in that ventricular model upstairs, and they're pink on the or pink or reddish color on the ventricular model. You know what I'm talking about? Those are networks of capillaries that are very, very permeable. They're very porous. They allow stuff in and out. Sitting on top of those beds of capillaries, you have a special kind of neural cell called an ependymal cell. And what they do is they filter fluids and stuff dissolved in the fluids out of the choroid plexus, which are the capillaries, and allow that fluid to drain into the ventricular system. Right? Each one of the ventricles has a set of its own choroid plexus and adjacent ependymal cells. And so this is really where you're making most of the cerebrospinal fluid. Does that answer your question? The other kind of plexus that we talked about were nerve plexuses. And they are just these huge interwoven branches of nerves that are coming together and splitting off.
Oh, okay.
There were 3 of them, right? We talked about the cervical plexus, we talked about the brachial plexus, and then we talked about the lumbosacral plexus, right? But that's really all you have to know.
Okay.
So the only thing we really need to know plexus is those 3 of them.
Yeah, those were the only 3 that we actually gave a name to. which one is in the neck, or which one is coming from the cervical. It's just there. There's really not much
So
out
is the cerebral spinal fluid made in the choroid plexus or in the, like, actual ventricle?
Same. That's the same thing. The choroid plexus is a region of the ventricle where you have those capillaries. So it is made by cells that are literally in the ventricle itself, right? So I wouldn't give you both of those answers simultaneously because they're both correct, right? You just gotta pick out the one from the lineup that is appropriate. So how are we on? So the one real chunk of physiology that we don't ever touch on in the lab is the action potentials. Do we feel okay about action potentials, or do we want to go over that stuff?
Yeah, because I had a couple questions on
Okay, so we'll start off simple, and there will be some of these questions as well. We've got a membrane, right? We've got outside the cell, and we've got inside the cell, right? Historically, I've done it from inside here and outside there, but I could very easily flip this around, right? So make sure you understand that it's not really top and bottom. It's outside the cell and inside the cell, because I could just as easily write in, out, right? So, now, for what it's worth, in, out. Most of your sodium is where? Out. Okay. Wherever you got sodium, you got chlorine. That's why blood is salty. What's the other thing over here?
calcium.
What's in here? Potassium. Protein we really don't care that much about because we're not moving it around. So this is less important. I mention it just because it helps you to understand how the overall charge distribution is accomplished. Now, I botched one of the questions on the study, or on the review questions. Did y'all see the disclaimer about that? That was my bad. That's still technically a legitimate question because even though most sodium is here, there is a little bit in here. It's the relative distribution that I've given you here. So, but that was my bad. I certainly wouldn't do that on the test. I just made a mistake. So, now what's that effect on charge distribution? More negative here, more negative here.
Bottom.
Negative on the inside, better. So, positive there. The result is a voltage across the membrane called the resting membrane potential. Right? About there. Whatever. So, not zero. Right? That's the critical piece of information. So, whenever I say a cell is a resting membrane potential, right, that is telling you that this is what it looks like. Okay? So when I say that it's starting off like this is what I mean. If you can do all of this, right, that's a good step forward to answering these questions. Now, what happens if we move chlorine over to here? It hyperpolarizes. Right? Negative charge to the negative side makes this more negative, this more positive. Right? Now, what's going to happen to this number if I hyperpolarize it? Yeah, it gets further from zero. Right? It gets more negative. Now, what I might do is I might graph this. I might have my electrodes somewhere in the membrane of the postsynaptic terminal, for example. And I'll say the cell's at rest. At this point, you move chlorine, or you move something. 60, 0, 100. What's going to happen to this line if I move chlorine and hyperpolarize? It's going to go down. So I might show you this graph, and I might say then which of the following ions might be moving at this point. Okay. Chlorine, What's the other one that will cause this to go down? Potassium. Because you move potassium to the positive side gets more positive, negative side gets more negative. This is hyperpolarizing. What's happening here?
Depolarized.
And then which ions will depolarize?
Sodium, calcium.
Yeah, sodium and calcium. Assuming they're moving down their concentration gradient. Okay. So how are we with this? Okay, so that's what's up?
It's they're moving away the numerical aspect of it, you know, it moves away from far away from auto 0.
Mhm.
That's what I need to get.
Yeah, basically.
Yeah. So with the first graph on there, with chlorine coming down.
Yep.
Yeah.
What about it?
Yeah, with chlorine going
Yes.
inside the cell makes it to be hyperpolarized. So
That's
we get
right.
away from 0.
That's correct. It gets
Okay.
more negative.
Okay. More negative.
Absolutely. Now this is presupposing that you are starting at resting potential. So let's say then that this comes back down. This is really the same thing that's happening here, but your starting point is different. And so I wouldn't call this hyperpolarizing. What would I call it? Repolarizing. Same ions that do this will do this section
And that
as
would
well.
take it back across the membrane.
Yeah. Right, so again, moving a potassium from in to out will do this, but moving potassium from in to out will do this as well. Right? Same thing. Does that make sense? Okay. So, all right. So this is just some random place in the cell. Now let's attribute this to specific locales within the cell. Let's get a little more complicated. So that was at the postsynaptic terminal. Right? Let's move now to the base of the axon. At the postsynaptic terminal, what channels were opening? Ligand-gated channels, right? That's what happens at the postsynaptic terminal. Now what we're going to do is we are going to go to the base of the axon where you have the trigger zone, right? Where we're going to initiate the actual action potential. Starting at rest. What's going to happen is now that we are depolarizing the postsynaptic terminal, those ions are going to drift to the other side of the cell where you have the base of the axon. And that means the base of the axon is slowly going to depolarize, right? Until it gets to a certain point, right? Right about here, this is known as your threshold potential. Right? At this point, what happens is it spikes up. So right here, you've opened your ligand-gated channels. What are you opening right there? Your voltage-gated channels. In this case, right, the only one we've talked about specifically were your voltage-gated sodium channels. Now, the next thing that happens is it's going to level off and then start to drop again. Right? You have opened another set of channels up here. Right? Ligand-gated or voltage-gated? Nope. Gotta be voltage-gated then.
How do you know?
Well, because this is what we talked about, right?
Right, I remember that, but like,
It's because we're in the presynaptic. We're in the
So, what happened, right? How you know is a different question. I mean, if you're asking how this was scientifically determined to be begin with. What they do is they use little teeny tiny electrodes that they actually stick on either side of the membrane. It's called a patch clamp. And then you can artificially open and close some of these channels, and you can artificially change the voltage across the membrane and see what happens. So there's a lot of experimental evidence to this effect. These were voltage-gated sodium channels. What we're doing here is, what's the name of this process? It's not depolarization, it's repolarization. And what do we move when we're repolarizing? Potassium. So these are voltage-gated potassium channels. So at this point, the voltage-gated sodium channel, right, when it spikes all the way up, the voltage-gated sodium channels close, and the voltage-gated potassium channels now open, right? And that causes sodium to go from in to out, causes your voltage to shoot way back down, right, until it actually levels off back at resting membrane potential. And this is our action potential, right? This wave is going to propagate all the way down the membrane. So, resting potential. You open up the ligand-gated channels at the postsynaptic terminal. Ions drift to the trigger zone and open up the sodium-gated, the voltage-gated sodium channels. You have a spike to zero and past, right? It actually reverses polarity in most cases. That opens the voltage-gated potassium channels, spikes way back down, and reestablishes resting potential.
Could it be voltage-gated chlorine channels too?
Theoretically, yes. In practice, the only thing that gets used is voltage-gated
So we will
potassium.
address chlorine.
Right. Where we talk about moving of chlorine is when you're talking about actions at the synapse. Right? What we said we use chlorine for were inhibitory signaling at the synapse. Okay. So, everybody know how to read this? So what I'm going to do is I'm going to have this graph. I'm going to label it A, B, C, D, and maybe even E. And then I'm just going to ask you questions. What's happening there? Where do you have sodium moving in? Where do you have potassium moving out? Which of these is repolarization? Which of these is depolarization? So on and so forth. All right. The only other thing is what Kayla was alluding to, which is the actual signaling event that's occurring at the postsynaptic terminal, right? So we got those ligand-gated channels, right? Neurotransmitter released into the cleft, binds the ligand-gated channel, opens them up. Depending on what channel it is, you move one ion or another, right? Two effects that you can have, right? You can take a resting cell at membrane potential, and you can spike it up, depolarizing, or hyperpolarizing. Or you can do it the other way. And then that would be hyperpolarizing. Right? This is at the postsynaptic terminal. Right? Is this inhibitory or excitatory? Inhibitory, right? Because it actually, at the same time that this is happening, right? If you look at the graph at the axonal hillock at the trigger zone, right? We said that what you want to do is get it to the threshold potential, right? But we're actually getting farther away from the threshold potential, right? And so that's actually inhibitory. That's going to be excitatory. Right? Alrighty. So that seems pretty good. We okay with that? What else can I do for you all? About 15 minutes. Well, excellent. I'm going to go eat lunch.
This is postsynaptic terminal, right? Post.
Yeah, this is here.
So you're
Threshold potential is a feature really of just the axonal hillock and anywhere that's got those voltage-gated channels. Because the threshold potential is the potential required to open up those voltage-gated sodium channels.
So are you only going to ask us about the axon and the posterior, or?
Well, I mean, where else would I ask about? I mean, I could ask about the middle of the axon, I guess. In the middle of the axon, you don't have, in the middle of the axon, right, you don't have that, you don't have that slow raise at first, right? Because this is relying on opening the ligand-gated channels to get you to threshold. In the middle of the axon, you're using one set of voltage-gated channels to open the next, right? And so in the middle of the axon, what it actually looks like is it just goes, right? So. Realistically, though, I mean, hyperpolarization, repolarization, depolarization. Can you identify them on the graph? Can you relate that to what ions would be moving theoretically?
So
And-
once you hit the trigger zone, it's voltage-gated.
That's right.
I'd have to know if it's sodium voltage-gated or potassium voltage-gated.
Well, yeah, because you got to know. But again, that leads back. Sodium is going to depolarize. Potassium is going to repolarize. And so, based on a graph like this, since you know this is depolarizing, it's got to be voltage-gated sodium channels. This has got to be voltage-gated potassium channels. There are other ways to do it, right? There are voltage-gated calcium channels that you'll talk about with the heart, presumably. Right? There are other ways of doing these things. Right? This is just, we're just cutting our teeth in preparation for stuff that is going to be way more obnoxious in the next semester. So.
So sympathetic and parasympathetic?
Yeah. What about it?
That's what I was
Okay.
Opposite.
General question about what they do, right? One of them's the fight or flight response. Which one's that? Sympathetic, right? Very easy question to that effect. The vagus nerve is going to be very important in terms of its role in the parasympathetic branch, and so I would definitely know that. There are other contributors to the parasympathetic branch. There we go. There are other contributors. There are a couple of other cranial nerves that contribute to the parasympathetic branch, but we're not worried about it. There are some of the spinal nerves down here that are contributory as well, some of the lumbosacral stuff. And then the sympathetic stuff is coming mostly out of the thoracic spinal nerves, right? So very easy questions to that effect. There will be one question that's about that obnoxious stuff about the architecture and the difference in the architecture between the somatic motor component, the difference between the somatic, the sympathetic nervous system, and the parasympathetic nervous system, right? All three of which are parts of the motor division of the peripheral nervous system. One thing I would do when you get to the exam, two sets of information at a minimum that I would just scrawl out on the back of the exam. Cranial nerves, name and number, maybe even sensory motor mixed if you really are wanting to get it done. The other thing I would do is just how we divide it up. We said the nervous system is split into the central nervous system and the peripheral nervous system. We didn't do anything else here really, although you could if you wanted to. We said we're going to divide this up into motor and sensory. We divided motor into somatic and into autonomic. We divided the autonomic into the sympathetic nervous system and the parasympathetic branch of the nervous system. We also divided sensory. What did we divide sensory at? General versus special. All right. So when I ask you if a neuron in the parasympathetic branch of the nervous system is motor or sensory, it's got to be motor. right? Because it's all branching off this motor cord. It's also peripheral. Right? So much easier to answer those questions if you just write that out. Okay, so now back to this one. We said that there were some major differences because of how this stuff is architectured, right? So one is just the number of motor neurons. This is generalized, right? Generally speaking, how many motor neurons are there in a somatic pathway? It's been a while since we looked at this. 1. We said, in fact, in the very beginning, that neurons are some of our longest cells because for the somatic pathway, I've got to have a neuron that erupts from the spinal cord, or I've got to have an axon that erupts from the spinal cord. It goes all the way down to the tippy tip of my foot. Right? 1 motor neuron in a lot of these somatic pathways. How many in the sympathetic? Do you remember? It's not 1. 2. 2 there as well. Right? This is where we get into the pre and the post-ganglionic neurons. Okay? And so you see similar architecture in both of these. Now, another thing that might be a testable unit is the body of the neuron. Right? Where's the body of the somatic motor neuron? We did this in the lab as well. You can actually see it in the lab. You know it? Yeah, it's in the gray matter. It's never in the white matter. What is the white matter? Myelinated axons. And so gray matter's got to be unmyelinated axons, neuronal bodies. Right? So bodies of my somatic motor neurons is in CNS. Usually. This is somatic. Now, we've got 2 different bodies to deal with. We've got a pre and a post. The pre, where's the body? Same as that one. Yes. Post, not in the CNS, in the PNS, right? But you got to be more specific. It's in one of those peripheral ganglia, right? In the case of the sympathetic nervous system, it would be in the paravertebral ganglia. And then not a paravertebral, but a terminal ganglia. Terminal meaning it's close to the organ that is ultimately going to be innervated by the autonomic pathway. Now, when I say that the body is in the CNS, though, a preganglionic neuron is not a central nervous system neuron because part of it is exiting out of the central nervous system. Right? So this is still a peripheral neuron, right? Even though its body is in the CNS. Often those questions might be right next to one another, right? So, now, let's throw in something else. Sensory, and we'll just do general. Where's the body of a sensory neuron? Nope. It's in a ganglia. What's the only other ganglia we know? Yeah, that's what I was looking for. Say it. The dorsal root ganglia. Right? Because these sensory neurons are those unipolar neurons that have that body right in the dorsal root ganglia. Often it's only one, but it can be more, so I'm not going to ask about that. Alrighty. So, thoracic, cervical and lumbosacral stuff.
What did you say was thoracic?
The sympathetic nervous system is originating out of spinal nerves in the thoracic region. Okay. This is out of the cervical region and out of some of the lumbosacral stuff. Okie dokie. So that's the highlights of the autonomic nervous system. They're highlights of the motor division really also. So what else can I do for you? I've got 3 minutes, 4 minutes. So what's the first thing that light passes through as it's going into the eye? The cornea. Cornea. Hanging over the cornea, you've got your eyelids. What's our term for an eyelid? Palpebra. Yeah, you can pick it out of a lineup, right? That's all that really matters on this multiple choice stuff. So, what's the pigmented layer?
Iris.
And what's the hole in the iris?
Pupil.
Pupil. What's back here?
Posterior cavity. Yeah, posterior cavity.
Yeah, we're not there yet.
Lens.
The lens.
What about the ciliary body?
Yeah, we're gonna. We're just following the path of the light, though. So, now what's this space in front of the iris posterior to the cornea? What's this space in here? The anterior chamber of the anterior cavity. This is kind of a mess. What's the space behind the iris immediately around the lens? The posterior chamber of the anterior cavity. Yeah, I know, that's where it gets confusing. And then back here,
So anything behind the lens is your posterior cavity?
yeah, basically.
Okay.
Yeah, okay. So what's the fluid here?
Your
We'll come back. We'll come back to that actually. So in the back of the eye, what's the first tissue that it's going to encounter? The retina. And so what's this part here? Very good. What's that part there? That's your blind spot. What's immediately behind the retina? The choroid coat. So anything not absorbed by the retina is absorbed by the pigmentation of the choroid. What's behind the choroid? The sclera. Now, there's a little indentation back here where most of the light gets focused on. That's the fovea centralis within the macula lutea. Okay, so half of the light between the palpebra, through the cornea, into the anterior chamber of the anterior cavity, through the pupil of the iris, into the posterior chamber of the anterior cavity, through the lens, right, ultimately transitioning into the posterior cavity, all the way back to the retina, and then if not by the retina, then into the choroid cavity. We've got this region here, that's the anterior chamber of the anterior cavity. What's that fluid? The aqueous humor. We've got this region here that's this. What's the region? What's the region immediately around the lens? The posterior chamber of the anterior cavity. What's the fluid in the posterior chamber of the AC? Nope. It's aqueous humor. And then we've got this region way back here. What's that fluid? That's the vitreous humor. Now we've got They transition into the sclera. We've got an enlargement here that's a muscle. What muscle is that? Yeah, the ciliary body or the ciliary muscle. You've got these processes hanging off that ultimately are attached to the lens. The ciliary body is then really, and this kind of is merged into the iris once you get forward enough. And then it's going to come back and it's going to merge in part with the choroid. The retina is going to just kind of appear out of nowhere somewhere about there. Okay. You know that you've got to have aqueous humor back here behind the iris because it's actually being made at these ciliary processes, right? And within the epithelia of the ciliary body. Aqueous humor circulates around the lens, forward, and then ultimately out through a little hole. All right. That seems pretty easy. We okay with that? There's a lot of information. We'll get there. 36 hours, right? More than that, 48 hours. Yeah, 48 hours. 48 hours. All right. So. Any other questions before we start with underside of material today? All right. How do we feel about the year?